Hello, hi, namaskara. My name is Kirtan and I am a second year medical student studying in a government medical college. Welcome to my channel Physiology at Once. Today I will be talking about the topic gastric secretion. It is one of the important topics in the GIT system, and in this video we will be talking about the functions of the stomach, the gastric juice, the factor stimulating and inhibiting the gastric acid secretion, and the process of the mechanism of the HCl secretion, the regulation of the gastric secretion. We will talk about the peptic ulcer in the next video. So let's start this video. Before starting this video, if you haven't subscribed to my channel, please subscribe because it's free, and also you can support my mission of making physiology easy and simple. You'll also get the Telegram channel link for the notes in the description. So let's start. What are the functions of the stomach? The anatomy of the stomach I will not be discussing here because it is in the anatomy chapter, in the abdomen part. So we will only talk about the functions of the stomach, the gastric juice and its secretion. So the functions of stomach. The first one is the temporary storage of food. As you know that stomach stores the food for a little amount of time before passing it to the small intestine. The next one is the grinding and mixing of the food. The food which is digested in the stomach is mixed properly and forms chyme. It is mixed with the HCl and the pepsin secretion of the stomach. The next one is the controlled emptying of the food. It delays the fast emptying of the food into the small intestine and makes it in a controlled rate. This ensures that the small intestine is well maintained and does not overexpand. The next function is the secretion of hydrocolic acid. The hydrocolic acid is important in the stomach, which helps in the killing of the microbes and also in the converting of the pepsinogen to the pepsin. This HCl also converts the ferric form into the ferrous form. So whenever there is a deficiency of HCl in the stomach, there will be a deficiency of iron and then cause iron deficiency anemia. The next one is the killing the microorganisms. This is due to the HCl secretion of the stomach. The next function is the activation of the pepsinogen to the pepsin. This is made possible due to the HCl secretion of the stomach, and this pepsin helps in the digestion of the proteins which is there in the stomach. The next one is it secretes intrinsic factor. This intrinsic factor helps in the absorption of the vitamin B12. The next one is the absorption of the water and lipid soluble substances. So now let's talk about the gastric juice. The gastric juice, 1 to 2.5 liters is produced per day. Its pH is almost 0.7 to 4. The composition of this gastric juice, it is again divided into inorganic constituents and the organic constituents, which was seen earlier in the saliva. The anions like Cl-, phosphate, sulfate and HCO3- and cations like H+, Na+, K+, calcium and magnesium are found in the inorganic constituents. In the organic constituents, pepsinogen, intrinsic factor, mucin, renin, carbonic anhydrase, etc. are present. The gastric secretion includes the secretion of the HCl, which is the important one, and the other major constituents such as synthesis and secretion of pepsinogen, mucus and intrinsic factor. So gastric secretion involves all of this. Mechanism of HCl secretion. Among all of this, the mechanism of HCl secretion is the most important one. So let's study this one. The HCl is secreted from the parietal cells of the stomach, which is found in the fundus region of the stomach. Fundus region of the stomach. The H+ secretion of the HCl takes place through the H+, K+, ATPase in the apical membrane of the parietal cells. So let's talk about this mechanism through a diagram. So this is the diagram of the gastric cell, that is the parietal cell. This is the gastric lumen and this is the interstitial fluid. So the main components of HCl is H+ and Cl-. So from where is the H+ produced? The H+ is produced due to the decomposition of the H2CO3, which again gives rise to HCO3- and the H+. This H+ is again exchanged with the K+ ion, and this H+ goes into the gastric lumen and fuses with the Cl- to form the HCl. So where does the Cl- come from? This Cl- is exchanged with the HCO3- which is produced here. So this Cl- goes through the parietal cell and comes out in the gastric lumen and fuses with the H+ to form the HCl. So whenever there is production of one HCl molecule due to the HCl production, there is one HCO3 molecule which goes to the interstitial fluid. So this H+ is exchanged with the K+ ion. So where the K plus ion comes from? So there is the Na plus K plus ATPase which is present on the parietal cell. The K plus ion exchanges with the Na plus ion and comes into the parietal cell and is again goes to the gastric lumen. From the gastric lumen, it is again exchanged with the H plus and again the K plus goes to the interstitial fluid. So this is a recycling of K plus process. So let's revise this again. The H2O3 decomposes to HCO3 minus and the H plus ion, which gives rise to the H plus ion, again which fuses with the Cl minus ion to form the HCl. So this is due to the H plus K plus ATPase. So there are two ATPases, Na plus K plus ATPase and the H plus K plus ATPase. So what is postprandial alkaline tide? So since you know that the increase in rate of HCl secretion, the level of HCO3 minus increases in the blood. So whenever there is one HCl production, there is HCO3 minus production in the interstitial fluid, that is the blood. So therefore, when the pH of the blood increases, the blood and the urine also become alkaline. So there is the alkaline tide, which is nothing but the postprandial alkaline tide. Prandial means after food. So postprandial alkaline tide. So now let's talk about the factors stimulating and inhibiting the gastric acid secretion. So the stimulating factors for the gastric acid secretion, they are acetylcholine. It is the hormone and it acts on the M3 cholinergic receptors. It increases the intracellular calcium. The next hormone is the gastrin. It acts on the gastrin receptors. It also increases the intracellular calcium. The next hormone is the histamine. It acts on the H2 receptors. It increases the intracellular cyclic AMP. And there is also mechanical and chemical factors. So when there is gastric distension or when the gastric walls expand due to the input of the food from the mouth, there is production that stimulates the gastric acid secretion. And chemical factors means when there is intake of hot and spicy food, there is the release of gastric acid secretion. So what are the inhibiting factors? The first one is the somatostatin. This is secreted from the D cells of the gastric mucosa. This is one of the important hormone which inhibits the production of the gastric acid. The next one is the pH of the gastric luminal content. So when the pH of the gastric luminal content decreases below 2, so when there is decrease of pH below 2 in the gastric lumen, there is again auto feedback, that is auto regulation of the gastric acid secretion. That is the negative feedback mechanism. The next one is the acidic chyme in the duodenum. Similarly, when the chyme in the duodenum becomes acidic, there is decrease in the gastric acid secretion. Now let's talk about the regulation of the gastric secretion. It occurs in three phases: the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase, it mainly involves the activities in the brain. It almost 30 to 50% of the gastric juice production occurs in this phase. It is elicited mainly by the smell, sight, thought, taste, and the chewing of the food. So these are the sensory stimuli for the activation of the cephalic phase. It is mainly mediated by the vagus nerve. This vagus nerve again has cholinergic fibers and the non-cholinergic fibers. So let's understand this by the flowchart. The vagus nerve has cholinergic and non-cholinergic fibers. These cholinergic fibers acts on the ECL cells, that is enterochromaffin cells, and the D cell. These enterochromaffin cells produce histamine, which stimulate the parietal cell and therefore increase the HCl secretion. Then the D cell produces the somatostatin, which is the inhibition factor and decreases the production of the HCl secretion. So this takes place to the acetylcholine hormone. The non-cholinergic fibers stimulates the G cell and produces gastrin. This gastrin hormone increases the activity of the parietal cell and the HCl secretion is increased. So the cholinergic fibers acts through the ECL cells and the D cell, and the non-cholinergic fibers acts through the G cell. Now the second phase is the gastric phase. This is the most important phase of the gastric acid secretion. It involves 50 to 60% of the gastric acid secretion. The main stimuli are the distension of the stomach or the expansion of the gastric walls and the products of the digestion, that is the peptides and the amino acid. So the distension of the stomach mainly acts to the central reflex and the local reflex. The central reflex is mediated by the vagus nerve. The local reflex is mediated by the acetylcholine hormone and the gastrin release by the G cells. The amino acid and peptides stimulate the G cells to the gastrin. So it also involves the gastrin cell release. So now let's revise the cephalic and gastric phase in a flowchart. In the cephalic phase stimuli like the sight, smell or chewing of the food activates the limbic system, which further activates hypothalamus and the vagal nerve. This vagal nerve acts to the enteric neurons and activates the G cell and produces gastrin, which further produces the increase in the H cell secretion through the parietal cell stimulation. And this enteric neuron also stimulates the E cell or the enterochromaffin cell, which produces the histamine, which again stimulates the parietal cell. So as we studied in the cephalic phase. In the gastric phase, peptides and the proteins and the distension of the stomach are the main stimuli. They act to the vagovagal reflex, that is by stimulating the vagal nerve again, and again they act to the G cell and the enteric neurons. The vagovagal reflex is nothing but that the afferent neurons are also the vagus nerve and the efferent neurons are also the vagus nerve. So now let's talk about the last phase of the gastric acid secretion, that is the intestinal phase. It involves only the 10% of the gastric acid production. The overall effect of this phase is the inhibition of the gastric acid production. So let's understand through this the flow chart. There is entry of chyme into the duodenum. So which I have written in red, they are the stimulating factors, and which I have written in green are the inhibiting factors. So the stimulating factors, there is distension of duodenum, which stimulates the vagovagal reflex, which I talk now. Then there is stimulation of duodenal endocrinal cells, which secretes the enteroxytocin. So this results in the stimulation of the H cell secretion from the parietal cells of the stomach. This is the initial phase of the intestinal phase. This is the stimulating phase. Then there is the activation of the enterogastric reflex. Enterogastric reflex is nothing but the negative feedback mechanism through which the parietal cells are decreased and there is decreased H cells production. Then there is release of the GI hormones like secretin, GAP, VIP, bulbogastron, enteroglucagon, etc. So this further results in the inhibition of the H cell secretion from the parietal cells of the stomach. This occurs in the later phase of the intestinal phase. So therefore the initial phase of the intestinal phase is the stimulating phase and the later phase of the intestinal phase is the inhibiting phase. So that's it for today, guys. If you found this video informative and useful, please like, share and comment, and also don't forget to subscribe. I will upload the next video about peptic ulcers. Thank you.